Okay, good afternoon. Good morning, everyone. So in the last class, we had looked at these two experiments. That was one of Messelson and Stahl's experiment, and the other one Tyler's experiment, where they were proving the point of semi-conservative method of DNA replication. So moving forward from there, now we'll actually come to the mechanism of DNA replication. How exactly does it happen? So there are several steps involved in the this mechanism. Again, at this stage or during this mechanism, there will be so many new terminologies, especially in terms of the enzyme names and certain specific structures, and we will have to remember all of it. So first step being the activation of deoxyribonucleotides, that is whatever they are present, they will get activated. Recognition of the initiation point from where the DNA replication has to start. Unwinding of DNA because it is present in the double helical structure, it will open up. Priming or formation of the RNA primer. So that is like a starting point for the replication process. Like it develops a kind of primer. Then base pairing and the assembly of the complementary strands. So actually the newly added base pairs will form complementary bonds with each other and they will form the complementary strand. Then conversion of these deoxyribonucleosides to from triphosphate to monophosphate residues, and then the formation of the DNA chains on the RNA primers, what is called as polymerization. So we'll see one by one each of these steps. So now the first step, that is activation of deoxyribonucleotides. There is too much of text, that is why a little different colors I have highlighted, and I will explain it step by step so it should be more clear. So first step, that is the activation of deoxyribonucleotides, monophosphates. So we have seen this in the earlier classes, that this is deoxyadenosine monophosphate, deoxythymine monophosphate, deoxycytosine monophosphate, and deoxyguanine monophosphate. So these are the four deoxyribonucleotide monophosphates which are present. They get converted into triphosphate, which are called as again deoxyadenosine triphosphate and so on, using the ATP as the phosphate molecule to gain the phosphate molecule. And this reaction is catalyzed by the phosphorylase enzyme, and this particular process is called as phosphorylation, which means addition of phosphate group. So that is the first stage. You might wonder where would this be available from. So obviously we know that there are nitrogenous bases and then there are phosphate molecules along with the sugar molecules, they will be joining, and this is a continuous process. So from ATP you continuously keep getting AMP or ADP, that is from adenosine diphosphate to adenosine triphosphate. It will break down, it will release energy, it will form ADP, that is adenosine diphosphate. It will again remove phosphate group, it will form adenosine monophosphate. So that is a chain of reaction. Again this AMP, that is adenosine monophosphate, will get utilized in the reaction and it will again form ATP, again that ATP will be broken down. So it is like a chain of cycle in different reactions, right? It's not just DNA replication, but all the metabolic processes within the body, they utilize ATP and they will form AMP. So that is the source of this deoxy AMP and so on. And same is the case with the other nucleotides. So from that from monophosphate, it will become triphosphate and it will be utilizing ATP. So ultimately it is utilizing the energy and with the help of enzyme called phosphorylase. So that is the first step of phosphorylation of deoxyribonucleotides from monophosphate to triphosphate. Now, the second step is the recognition of the initiation point. Now, DNA replication is initiated at a defined sequence of the nucleotides. And what does this defined sequence means? It is called as initiation codon, or which is also called as origin of replication. In short, it is ORIC. So initiation codon, now we know it, it is ATG. That is the initiation codon. So it's like a scanning mechanism over the DNA fragment. So it will go and bind to the what we call as ATG, which is the first initiating codon or starting codon. It is along with that, there will be certain specific sequence. So this defined sequence does not only mean the ATG as such, but along with that, there will be another. So in a way, what you can say is there are two start site, but not literally when I say so, because one is the what we call as initiation point, and one is the actual starting point of the gene. Yeah, these two are two different things. When we talk of the initiation point, it is before ATG, and when we talk of the actual start point of the gene, we mean ATG itself. So before ATG, there will be initiation point, and this particular sequence will be somewhere around 100 to 200 base pairs. Can be more at times depending on what gene, what protein complex, and so on. So that is the initiation point of this replication process. Specific initiator protein, which is called as DNA, will recognize the initiation point and binds to it. Now again, this protein will have specific recognition site for the DNA molecule of that particular gene. And before every gene, most of the initiation point will have a kind of common sequence, but not so common that they are same for all the genes. They have subtle differences amongst the nucleotides of those initiation point sequence, and that is when this DNA molecule, which is the initiator protein of the replication process, it will go and bind to that specific sequence. So after that, after the DNA binds to the initiation point or at the ORIC, which is the origin of replication, unwinding of DNA starts happening. So once the this initiator protein has bound to the two strands of the double helix, that will be taken as the unwinding point. And then, so the initiator protein will denature the DNA after binding to the origin of replication with the help of ATP. Now, obviously, to break that hydrogen bond, it will need certain energy, and that is how it will take that energy from the ATP. And the bacterial histone-like protein, which is called as HU. This is not actually the bacterial protein, but it is bacterial protein-like structure. So that is what bacterial histone-like protein, which is called as HU. So this DNA along with this bacterial histone-like protein will cause the denaturation of the DNA. It will take the energy from the ATP and it will cause the unwinding. So the two hexamers of the helicase enzyme, which is DNA will bind to each strand of the unwound DNA. So once that opening has been created by the DNA and HU, DNA which is another protein, along with the helicase, will go and bind to that unwound DNA, which is very close to DNA. Now, if I have to show you this particular part, I will just take a help of the board. 1 minute. So let's say now I won't be able to draw the. Oh no, let me try it. Yeah. I may not be so good in drawing double helix, but let's say. Yeah, this is the DNA. right? And then we have, let's say, a certain sequence which is ATG here. And this is the starting codon of the gene. Before that, this particular whole of this sequence, let's say, right, all of this sequence, this is the initiator point, or what we also call as ORIC, origin of replication, right? ORIC. Now, DNA A will come and bind. Let's say this one is DNA A. Now, along with this, there will be HU, which is bacterial helicase-like protein. Now, once there are hydrogen bonds in between the nucleotides, right? So all of these will be the hydrogen bonds between two nucleotides. They will be broken completely with the help of ATP. So it will take the help of ATP, which will hydrolyze the ATP. It will form ADP plus phosphate. Energy will be released. That energy will be utilized by this DNA to remove these bonds, so to break the bonds. So that way, these two DNA strands, they are now separated. Now, next thing that happens here is another enzyme comes, which is DNA B. You usually write this short DNA B, which is another enzyme. It will bind along with the helicase. Helicase is another enzyme. It is ultimately means helix. Ase means breakdown. The enzyme which breaks down the double helix. So what these two will do is they will go and bind next to DNA A. It will keep this particular two strands separate so that they don't merge again. Because otherwise, you know that within the cell, within the DNA molecule, there is a lot of what we call as already lot of energy. The two of the nucleotide base pairs, they are very close to each other within the DNA strand, so they might form the hydrogen bond again. So to avoid that, this helicase is bound there so that it does not allow these two strands to fall back again to their hydrogen bond. So two hexamers of helicase enzyme, two hexamers as in it is present in the form of hexamer, that is 6 molecules, and 2 of such molecules of the helicase and DNA B will bind to the each strand of the unwound DNA. I did a mistake there. It is the helicase itself is DNA B, which is the other name. So it will bind to each strand of the unwound DNA close to DNA A. Now DNA B along with the third molecule, which is DNA C, will unwind the DNA bidirectionally and will create a replication force. So that means third molecule will come in, it will keep moving forward and it will keep unwinding whole of these DNA strands, and then it will keep it just like that until the next reaction happens. Any question here so far?
So,
yes.
So DNA C is also a type of helicase, or only DNA B is?
So in a way they will all function as helicase, because at the end they are all opening up the replication forks, or what we call as DNA strands or DNA helix, right? So there are specific actions of each of these, but we are not studying that. So that's why it might sound like all three are doing similar function. So ideally what happens is this DNA A will go and bind. It will break open the one of the nucleotide because it is taking the help of ATP as an energy. So the moment it has broken that bond, so it will expose, it will invite DNA B to come and sit here. So DNA B will kind of unwound the DNA. So it's kind of in spiral helix. So it will kind of unwound and then DNA-C will come there and it will break open the further what you say, the hydrogen bonds between the DNA strand. So individually they all have a little of their action, but as of now here it all looks similar. So in a way you can call all of them as a helicase type of enzymes, but the action they are little different from each other.
So A, B, and C all are three different types of helicases.
Yeah, not, you cannot say that. That is what I am trying to say. The actions might look similar, so in a way you can call them so, but that is not the right way of calling. So the simplest answer to your question would be no.
Oh, okay. So only DNA B is a helicase.
Yeah.
Okay.
So I hope this first part is clear to you all. We'll move on to the next step. I will show you whole of this process, though that process does not completely explain each and every minute details like this. But yeah, that video will be kind of revision to whole of this process. So first step, activation from monophosphate to triphosphate of the nucleotides, then recognition of initiation point, that is that identified particular DNA A protein, which is the initiator protein, will go and identify the origin of replication, which is kind of before the actual gene sequence, somewhere around 100 to 200 base pair, depending on which DNA and what gene it is. And then next step will be the unwinding of the DNA, two strands, with the help of helicase enzyme.
So
Yes.
So the origin of replication and the initiation codon, are they the same or are they different?
They are different.
So in origin of replication is the first one, and the initiation codon is the second one.
Correct.
And sir, the 100 to 200 or more base pairs, is that the nucleotides in between them?
No, that is what I drew. Before ATG, there are few base pairs which we call as this origin of replication.
Okay, so all the 100 to 200 base pairs are called origin of replication.
Yeah, whatever that number is.
Okay.
Okay, so these are the three steps. Now, this unwinding, that means whatever DNA A, B, C together they have created, will create a tension on the two strand. It's simple thing, like if you have ever taken a thread and you try to unwind it and you keep it like this, it will again fall back, right, if you leave it. So this tension is released by the enzyme which is called as topoisomerase 1 and 2. So there are again another type of enzymes which are called as topoisomerase. So they will release the tension. Now how exactly, that we need not discuss here, because again that is a long chain of protein complex reaction which is involved. But if at all you are interested, I can share the resources with you later, so you can go through them shortly. So that topoisomerase 1 and 2 together will release the tension from the two strands which are unwound from the parent DNA molecule, and then the two strands of the DNA will start separating further by breakdown of the hydrogen bond, so that they can act as a template for the synthesis of the new strand. So from the action of this DNA A, B, C, there is a gap created. So the replication force, the two DNA strands which were in double helix, that will just open up a bit, right? But then it has to unwound completely. So to do that, the topoisomerase 1 and 2, they come into picture. They will release the tension from those two strands. So it's ultimately like how you make like a particular strand of the thread, you unwind it and you literally like kind of squeeze it through your hand. So that's how the tension gets released. Something similar happens in terms of this action of topoisomerase 1 and 2, right? But again, that is a protein complex mechanism. So that is why it need not be so simple, like we just squeeze this strand and it becomes straight separate. There is obviously some breaking and forming of the bond, and then it again releases the tension. So that is the general action of topoisomerase 1 and 2. Then the fourth step in the mechanism of DNA replication is the priming, or what is also called as formation of RNA primer. Now this terminology, primer or priming, will come quite often throughout the process, because primer, it refers to a short nucleotide sequence, right? Can be for DNA, can be for RNA, but primer is a relatively common terminology. In this case, we are specifically talking of RNA primer. So the enzyme, again another enzyme, yeah, DNA directed RNA polymerase. That means this is actually RNA polymerase, but it is directed by a DNA molecule. That means its action is controlled by the DNA. So that is why DNA directed RNA polymerase. RNA polymerases, they are used in the synthesis of RNA. So you might wonder, what are they doing in the DNA replication process? So just hold on to this particular fact. So DNA directed RNA polymerase synthesizes a short priming strand of RNA called as RNA primer on a DNA template. So again, we have identified the origin of replication. At that origin of replication, at the starting point, not at the start codon, at the origin of replication, DNA directed RNA polymerase will go and bind and will create a complementary RNA strand to the DNA template. Yeah, and this enzyme is also called as primase because it creates the primer on the DNA strand. Now this RNA primer is a short strand of RNA formed on a DNA template. Now why this is required? Because the DNA polymerase itself cannot initiate the DNA synthesis of a new DNA strand. It's something like, if you have to build something, you need some reference structure to build on, right? Reference structure does not mean the template, but to start that process forward, right? Like if you want to do something, I need to show it to you to do something, right? So that is what exactly the RNA polymerase does. So it will create a small primer at the origin of replication with the help of DNA directed RNA polymerase. So once that primer is created, DNA polymerase will come into picture, and from there it will start synthesizing the what we call as the further replication or further duplication of the DNA molecule. So DNA polymerase itself cannot initiate the DNA synthesis of the new DNA strand, but it can polymerize the growth of DNA chain. Polymerize the growth of DNA chain means it can add on to the nucleotides in sequence complementary to the original DNA template, which is in sequence or in further continuation from the RNA primer. If the RNA primer is not there, DNA polymerase will not be able to function. So the moment RNA primer is being formed with the help of DNA directed RNA polymerase can come there. It can start the polymerization reaction, and then this DNA primase, or this simple primase, what we know as, along with the DNA helicase, together they form this primosome. So DNA helicase, that means from the previous step, that unwinding DNA B protein or enzyme, and the DNA primase, they form this complex. So we are looking at step by step, right? But when it actually happens, these processes, they are so fast, within fraction of seconds you will have quite a lot of process finished already, right? So that is when we are actually looking at step by step. But otherwise, all these processes, they happen simultaneously in a fraction of second. You can imagine how fast they would be. And that is when you might wonder, how come the DNA helicase is coming in contact with the DNA primase, right? So that is when I'm saying like because it is so fast that these processes happens in a very short span of time. That is when these two molecules they are together, that is primase and helicase, and together they are called as primosome. Again, any question in this step? Is this part clear? Or if you have any question, you can ask.
So, this begins from the origin of replication or the initiation codon. This is from the origin of replication, right? So from the first nucleotide.
Yes, not of the gene, but of that gene template, whole of the DNA template. So like I had drawn on the board, origin of replication is before ATG. That's where it will all start.
Okay.
Okay, so that was the fourth step. Then the next step is the base pairing, or what we call as assembly of complementary strands. Now this is a simpler step. So we know that there are deoxyribonucleoside triphosphate. They will pair with the appropriate nitrogenous bases of the template DNA strand, and this particular reaction is facilitated by the DNA polymerase 3. So there are three types of DNA polymerases, 1, 2, and 3. This action of addition of base pairing on the RNA primer or in continuation to RNA primer is carried by DNA polymerase 3 specifically. So that is what is base pairing or assembly of this complementary strand. So that is the action of DNA polymerase 3. Now in continuation, what happens is, now simultaneously again we, like in the first step, what we had seen was the from monophosphate it had become triphosphate, right? Now what we have added is actually triphosphate. So deoxyribonucleoside triphosphate molecules, they will be there on pairing with the nitrogenous basis of the template strand. So both there is a continuous strand. In like in complement to it, there will be respective nucleotides, and these nucleotides, they are in triphosphate form. So they will set free a pyrophosphate, that means double phosphate molecule, and it will break down that phosphate separately, that two molecules of phosphate together, and the remaining let's say nucleoside monophosphate, right? Monophosphate means only one phosphate will remain with the nitrogenous base and the sugar molecule, and the other two, which is called as pyrophosphate together, they will be release free, and it will change into deoxyribonucleotides. Okay, from nucleoside triphosphate, it will get converted into deoxyribonucleotides. Now the enzyme pyrophosphatase, again pyrophosphate, and ase means breakdown. So this last ASE will mostly stand for breakdown. So pyrophosphatase, that means the enzyme which breaks down the pyrophosphate, will hydrolyze the pyrophosphate molecule into inorganic phosphate molecule. That is what is like individual phosphate, which is always denoted by Pi, which is inorganic phosphate, and this way it will again release the energy. So whatever energy was used earlier to form triphosphate, it is again released back at the end of this process after the nucleotides they are attached to the template strand. Again, any question in this particular process? So you might wonder why it is again converting from earlier it was monophosphate to triphosphate, and again it goes back to from triphosphate to monophosphate, because these processes or most of these biochemical processes, they will need certain energy. So that means the energy flow has to be continuous, either in the form of consumption or release. So that cycle has to continue, and that is why that change of reaction keeps on happening within a cell. So that is the conversion of this deoxyribonucleoside triphosphate to monophosphate molecules. Now, in the next step, the formation of new DNA chains that happens on the RNA primers, what we generally call as polymerization process. So, polymerization of DNA or polymerization of nucleotides will happen. So, the energy released by the breakdown of this pyrophosphate molecule is utilized in joining the adjacent nucleotides to form the polynucleotide chain. Now, so far, only the complementary bases they have attached. The sugar and phosphate backbone molecule of the newly formed strand is not attached yet, right? So that attachment of the adjacent nucleotides happens with the help of energy which is released by the pyrophosphate breakdown. And the process is catalyzed by the DNA polymerase 3 again, right? So addition of the nucleotide to the new strand as well as the formation of the sugar phosphate backbone happens with the help of DNA polymerase 3, and this specifically require manganese and magnesium ion for this DNA polymerase to function, because they act as a catalyst for this enzyme, that is DNA polymerase 3. So the new strands of DNA are formed in 5' to 3' direction on 3' to 5' template, right? It's obvious. Like, let me show you that something came on the top. Where is it? Not this. Where is it? Yeah. So the new strands, they will be formed on 3' to 5' strand, this blue strand, in the direction of 5' to 3', right? So that is what is being said there. Where is it? Here. Yeah. So the new strands of DNA, they are formed in the direction which is 5' to 3', but on the strand which is 3' to 5' template by the addition of deoxyribonucleotides to the 3' end, because it is going towards the 3' end. So at the 3' end of the RNA primer, it will always keep adding. So again, I will go back to this diagram. So it will start, let's say this was the origin of replication, and it will start from here. So first part will be RNA primer. The nucleotide bases will be always added to 3' end. So it will go in the 3' direction, and then it will keep adding the nucleotides on this 3' end. So that is what is being said there. So all these deoxyribonucleotides, they will be always added to the 3' end of the RNA primer. Now, the addition is affected, or means it's affected means it is controlled by the DNA polymerase 3 in presence of ATP. Again, that is a bond formation process. So for that bond formation, there will be energy required, which will be utilized from the ATP source. Once the synthesis of DNA strand has been initiated, it proceeds continuously, keeping the pace with the unwinding of the DNA at the replication. Now, like I said, this process is kind of simultaneous. So there will be whole long of DNA molecule. Now, when there is so much long of DNA molecule, it is not that two strands they separate completely and they stay as it is. This DNA A, B, C, and helicase and all those enzymes and even topoisomerase, they will keep running along the DNA molecule. And simultaneously, this DNA polymerase and DNA directed RNA polymerase, they will also keep running along the length of the DNA. So simultaneously, as and when these nucleotides are added, it is following the steps of unwinding of the DNA. So DNA is unwound, and immediately behind it, the DNA polymerase will keep adding the this nucleotide molecules, and that way the reaction continues at the replication fork. Again, any question in this process? Too many steps, too many names to remember, but as I said, we will have to remember all of them. So I assume this part is clear. Again, there are quite few confusing questions which are being asked based on this whole of this DNA replication process, right? So we will try to solve some of the question maybe sometime at the end of this chapter. because that will give us more and more idea to or scope to think about these processes.
So
Yes.
Deoxyribonucleoside triphosphate, it's the same as the normal structure, but the two extra phosphates are attached to the sugar.
Correct.
Okay.
You are asking about this triphosphate, right?
Yes, sir.
Correct. Okay, so not this one, I guess. We will watch this video. I hope this is the same video. Let me check. Yeah. No, this is on the unwinding DNA packaging. I don't know why did I put it. No, here it is. The process. Yeah. Okay, let me show you this video. So this is like a three-dimensional representation. We can have a look at this video. Some facts may be incomplete in this, but let's try to watch this video.
In this animation, we'll see the remarkable way our DNA is tightly packed up so that 6 feet of this long molecule fits into the microscopic nucleus of every cell. The process starts when DNA is wrapped around special protein molecules called histones.
Okay, sorry, I must mention that first part is the wrapping of these DNA molecules, so it is not related to the DNA replication, but how the chromosomes they are present, and then when they separate, they will show the kind of metaphase, anaphase, and so on, and then from there they will go to the DNA replication part. So this is in continuation.
Combined loop of DNA and protein is called a nucleosome. Next, the nucleosomes are packaged into a thread. The end result is a fiber known as chromatin. This fiber is then looped and coiled yet again. Leading finally to the familiar shapes known as chromosomes, which can be seen in the nucleus of dividing cells. Chromosomes are not always present. They form around the time cells divide when the two copies of the cell's DNA need to be separated.
When DNA replicates, its strands are separated by the enzyme helicase. Single-stranded DNA binding proteins keep the strands from re-annealing. One DNA strand encodes the leading strand, which forms from its 5' to its 3' end, using DNA polymerase 3. No problem here, but the lagging strand presents problems.
So this part we are yet to look at. Yeah. So this whole thing was just to show you. There is another video which we will watch, which is also a small video. So there that will also give more clarity as well. So this part, if you don't understand, you can skip it because we are going to study it. This specifically the lagging strand part.
Has to form from 5' to 3' too. It forms in pieces called Okazaki fragments. First, an RNA primase lays down an RNA primer. Then, DNA polymerase 3 lays down new DNA. The process repeats again and again. DNA polymerase 1 replaces the RNA primers with DNA. Finally, DNA ligase links the Okazaki fragments.
Okay, so that is like a short overview of whole of this process. You can see how many steps they have skipped, which is quite a lot. Let's have a look at this video, which also talks about the same thing. Again, a three-dimensional view. Maybe more we watch something like this, we'll get a little more clarity in terms of visualizing them.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand. And wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork.
So again, just to mention it here, you remember we spoke of the hexamers of 2 hexamers of helicase. Now you would imagine why it would need so many hexamers or 2 hexamers, which is like almost 12 molecules of helicase to separate the DNA strand. So that is just because of it's the structural requirement to unwound the DNA and create a replication fork.
The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called Dna Polymerase binds to the primer and will make the new strand of DNA. Dna polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
Okay, so I hope this process is clear to you. Do you have any question here so far? We will except the part where the video is talking about leading and lagging strand, that we have not seen. But have you generally understood the process of the DNA replication?
Sir.
Yes.
So when the RNA primase comes, which one is the one that adds the RNA nucleotides?
Huh?
So like it adds the RNA nucleotide. So those have uracil and those components, right?
Correct.
So and afterwards they have to be removed because they have uracil and those components.
Yes.
Can't directly the DNA counterparts be added. So then you just have to connect with DNA ligase.
That is where the question was, because DNA polymerase cannot function on its own to.
Yes, sir. So I'm saying that before DNA, before DNA polymerase comes to build the chain, what was that again? RNA primase, I think.
RNA polymerase.
Yeah, okay.
DNA directed RNA polymerase.
Yes, sir. Okay. RNA polymerase comes and it puts in some small section RNA counterparts, right? So can't something like DNA polymerase come and like, okay, it can't come. Like something else comes that puts the DNA counterparts. So you don't have to remove them and attach them again later.
See,
I mean, isn't that a redundant process?
Maybe it is redundant process, but then see, all these molecules, when we talk of enzymes or proteins and so on, they were part of evolutionary process, right? So you like something must have happened in a cell that it led to the synthesis of these molecules and the process like this. So one thing that I know of is each of the protein molecule has a specific receptor site, right? So the DNA polymerase, I know it cannot start the new process, right? And what you are thinking, what you are saying is like instead of RNA polymerase coming and adding those RNA ribonucleotides, it can directly add deoxyribonucleotide, or some other enzyme will take over the RNA polymerase function and do that same function. So it does not have to repeat the removal and again adding the DNA nucleotides, right? But in that process, what happens is the primer is most essential. DNA polymerase, when it acts for the synthesis on a new strand, it will check for the RNA primer. If it checks on the DNA primer, that means it can start again from anywhere, right? Normally the two DNA strands which are there, that also can act as a DNA initiating start site, right? Because your DNA polymerase or any other, for that matter, any newly formed protein molecule, whichever is starting the DNA replication process, that will recognize in the DNA strand itself anywhere. So that is when, to make that process very specific to the DNA replication fork, which has been unwound and kept it open, that's where the position RNA primer will go and bind. So that is recognized by the DNA polymerase, and from there only it can start synthesis. So that is one of the what we call as identity card of the DNA polymerase to start the replication process. Did you get it?
Yeah, sir.
So it is not like these processes like they just happen because they have been there. No, for each and every this process, there is always some explanation. We may be aware sometimes, we may not be aware sometimes, but there is always some explanation. Any other question? Okay, so if not, then we will stop here for today. Tomorrow is Saturday. We will have a class for sure. Tomorrow I don't have any meetings or anything. I hope there is just this. What is it? Tomorrow is Saturday. So let me stop recording.